afternoon. I'm Dr. Jen Kappa, and I'm going to talk to you about oxidative phosphorylation. So as we continue our journey through the process of the synthesis of ATP or adenosine triphosphate, which is considered the energy molecule of the cell, we are now at the last set of reactions that we refer to as oxidative phosphorylation. If we look real quickly at our overall equation for aerobic cellular respiration, we have glucose plus oxygen goes to carbon dioxide plus water plus ATP. As we've seen previously, the glucose is broke down during glycolysis. This is what this literally means, is the splitting of glucose. We saw that the 6 carbons from our glucose molecule have been released as carbon dioxide during both pyruvate oxidation and during the Krebs cycle, also known as the citric acid cycle. So now we're finally going to talk about the rest of the components during the reaction that leads to the production of ATP. The oxygen is going to be reduced during oxidative phosphorylation. The water is going to be produced, so what's going to occur is the oxygen is actually going to be transformed into water during oxidative phosphorylation, and the majority of the ATP is going to be produced during oxidative phosphorylation. All right, so if we look real quickly here, what we're looking at is a structure called the electron transport chain. Also known as the ETC. And what we see here on the y-axis, what we have is free energy relative to oxygen. The unit on our energy is kilocalories per mole. And what we have here is the electron transport chain. The electron transport chain consists of protein complexes that contain enzymatic regions that allow for the oxidation of NADH and FADH2, which were produced during our previous sets of reactions leading up to oxidative phosphorylation. And the complete breakdown of glucose, there was the production of our electron carriers, NADH, FADH2, which will now be utilized in the electron transport chain for the production of ATP. We have complex 1, complex 2, coenzyme Q, complex 3, cytochrome C, and complex 4. What we're seeing here on this graph is the free energy in kilocalories per mole is that we have this gradual drop in energy as the electrons from our electron carriers is being passed from enzymatic protein complex to protein complex. And we have this slow drop in free energy. And what this allows the cell to do is harvest incremental amounts of energy rather than one large burst of energy, which would be too much for the cell to actually capture all of that energy. So this is a much more efficient way of being able to capture the majority of the energy from the electrons that our electron carriers are donating to the electron transport chain to allow for the uses of that energy to synthesize ATP. What we see down here is that the least electronegative is up here in our NADH, right? NADH is an electron carrier. It's going to freely give up the electron to the electron transport chain, meaning that it's not that electronegative. It's not that greedy. It's not holding those electrons super close to itself. Whereas the most electronegative in this scenario, and in our cells, in our mitochondria, is 1/2 oxygen. So if we look at this portion right here, and I'm going to erase this equation now, but we'll come back to it, and we'll account for everything at the end. If we have 2 electrons plus 2 hydrogens plus 1/2 O2, this is going to give us a water molecule. So I just erased the equation, but I said that the oxygen and the water were going to be utilized during this set of reactions that occur in our series of aerobic cellular respiration, or during oxidative phosphorylation. Here's our oxygen, here's our water, okay? So if we look at what's actually going on here, is proteins are pumped, or proteins pump hydrogens from the mitochondrial matrix to the intermembrane space. So let's look at our mitochondria again. We have our outer membrane, we have an invaginated inner membrane, we have our matrix, and we have the intermembrane space. Inter, you'll recall, means between. So this is between our 2 membranes. The Krebs cycle is occurring within the mitochondrial matrix. And if we zoom in, the electron transport chain, real quick, let me show you where this is occupying space in the mitochondria, is sitting on this inner membrane. And if we zoom in here to the electron transport chain and grab a little bit of the matrix and a little bit of the intermembrane space, and I'm going to draw a really superficial inner membrane here, what we have is the electron transport chain sitting in our membrane. And here's the matrix. Here's the intermembrane space. And what we have is the NADH produced in the Krebs cycle being oxidized to NAD+. And in this oxidation step, what's occurring is the electrons are being passed down the electron transport chain, ultimately falling to 1/2 O2, which again is going to generate the water within the reaction. And what's happening is these protein complexes are pumping hydrogen across the inner membrane into the intermembrane space, which is creating this high hydrogen gradient in the intermembrane space. And we have few hydrogens down here in the matrix. So we have, I'm going to just use dots to represent the hydrogen ions and protons here relative to one another. So we're using the electron transport chain. The actual point of the electron transport chain is to harvest the energy to be able to push the protons against their concentration gradient. So we've already talked about this when we talked about membranes and we talked about diffusion, is that in most cases in the cell we use ATP in order to push molecules, ions, whatever we're talking about within the cell against their concentration gradient. To go against the concentration gradient, we must use energy. It's an energy-intensive process. And in the majority of cases, we use ATP to do that. But in this case, we're generating ATP, and therefore we're not going to use ATP to make ATP. Instead, we're going to use the energy in the incremental drops through the electron transport chain. The energy from the electrons is going to be used to pump the hydrogens against their concentration gradient, and that concentration gradient is what's going to be used to generate ATP. So then, the protons are going to move back across the membrane, passing through a protein complex called ATP synthase. This is a very rudimentary drawing. I will show you a much better picture. But what happens is the hydrogens fall down their concentration gradient. So this is not going to use energy because they're going down the gradient, not against the gradient. And what's going to happen is ADP plus inorganic phosphate is going to be synthesized into ATP on the protein complex that we call ATP synthase. So ATP synthase uses the protons to drive the phosphorylation of ATP. And we call it a phosphorylation reaction because ADP plus inorganic phosphate makes ATP, so adenosine di-2-phosphate is going to be synthesized into adenine triphosphate, and that is a phosphorylation reaction because we are adding a phosphate group to ATP. And this is an example of what we call chemiosmosis, using the energy of the gradient to drive cellular work. Thank you very much for watching. My time is definitely up. There's a lot more information here, but that is a quick snippet